applications. All the things that are explained by this Dr. Ganesh Kokate sir. And he's also used to give the on-site demo, application support, workshops on handling of software and its implementation. And he also doing presentation of basics of HPLC and its applications, basics of LCMS and its different application depending on customer requirement. And he also focused on specialized detectors, including RID, fluorescence, ELSD, etc. And he also doing the demo of gel permission chromatography. So with this brief introduction, I request to Dr. Usmani sir to welcome the Dr. Ganesh Kokate sir. So I request to Kokate sir, please accept the floral bouquet. And now I request to Kokate sir to start out the session.
Yeah, thank you very much. Such a warm welcome. It's obviously it's a visual, but yeah, it's a great welcome. I'm feeling that I'm there in your college or like that. So thank you very much, sir. And thank you very much for such a brief introduction as well. So probably I'm not going to waste a single minute because a very stipulated time is there, I know. And I need to cover a lot of things in today's session. So just a moment, I will share the screen at first and we will move directly. So if you have any concerns about HPLC or the concept of HPLC, so what I will do, I will just start the presentation from the basics of HPLC. And if you have any doubts or anything in your mind, please feel free to just ask whenever it is possible, or maybe we'll keep at least 5 to 10 minutes question answer session at the end of my presentation. So as sir already told me that Dr. Salvi sir already mentioned that I'm working in a Spinco Biotech as a senior application specialist since last 5 years. But before that, I was in pharmaceutical industries, mainly connected with USV, Cipla, Torrent, Heikel. So I have experience of the pharma as well, almost more than 15 to 16 years in the pharma. So whatever question if you want to ask, it's almost more than 70 participants are there. So it's a big platform. It's free to ask. So today I'm going to cover the concept of and the scope of the HPLC. Mainly we're dealing with just a moment. It's better to have the pointer. Yeah. So I'm going to cover the concept and scope of the HPLC. Apart from that, how the separation has been taken place, what is the actual mechanism of separation, and what are the different modes of the separation. So yeah, everybody knows about this concept of the HPLC. In this session of, you may say, what are the different terminology or what is the concept basically in the chromatography. I have also attended the previous session. Almost sir explained what is the concept of the separation and everything. I really glad to have that session as well today. So in the chromatography itself, in the analytical method, where we need to separate and quantify of any targeted molecule or any targeted sample from the matrix, and that is the main purpose of the chromatography. So if obviously I'm not going in detail about the theoretical part of the chromatography today, but apart from the chromatography, there are GC as well as the supercritical fluid chromatography. But today I'm going to focus only on the liquid chromatography. So in the liquid chromatography, why we are preferring HPLC? So in the category of HPLC, if you see, there are different concept basically all together. So if I want to say, if we prefer HPLC, the first concept coming in the mind is a high resolution. Okay, the resolution, what is the resolution, why it's required? So I am going to cover that as well in coming slides. But yeah, if you want a high sensitivity up to the ppb or ppm level, so parts per million or parts per billion, this HPLC will work depend on the sensitivity of the molecule to that particular detector. So today I will cover detector as well. And yeah, it's a good repeatability. So example, if I analyze the sample today, and as you all from the pharma background, so in the pharmaceutical organizations or any other organization, the first concept is about the repeatability. And if the repeatability will start, this is the most important phenomena basically when you are talking about the quantification or any kind of analysis. So small sample size. For the analysis of the sample, we required hardly the sample in ppm, so it's in the milligram, not required in the sample of a gram or like that. So sample size is very small. Apart from that, there is no drastic conditions which are required some pipe of gas pipe or everything. So it's not required as such. Only the moderate analytical conditions are enough for the HPLC, and the sample is not going to vaporize like JC. So that sample can be used for the Azure analysis as well. And the fractionation is also possible with the help of semi-preparative or analytical. When you are loading analytical samples, example 100 ppm or maybe 500 ppm. So at that time, we are also able to fractionate the sample and purify it with the help of very small quantity. I'm not telling about the collection of purifying about the grams or mg. So just getting the fraction to identify. And obviously the last but not the least, it's a non-destructive. So what are the scope, what are the different scope of the HPLC? Mainly we are using this HPLC in the field of starting from pharma to the clinical medicine. Normally there are different types of mixtures. Example, if I am talking about the industrial chemistry, obviously some of the surfactants, dyes, polymers, plastics. I think some noise is coming from background. Just a minute. Okay, fine. So in the case of pharma, need to check with if you want to do the analysis of some antibiotics or sedatives or maybe some steroids, some of the drug molecules. So all those kind of the analysis we need to check with the help of HPLC. In the field of food sector, obviously now in food sector, health sector is going a huge since last 3 to 4 years. So in food sector, normally toxin or maybe some additives or maybe some antibacterial activities we need to check with the help of the HPLC. We also checking with amino acids or maybe fatty acids analysis with the help of different detectors, coloring agents. So these are some typical analysis which are possible with the help of HPLC in the sector of food. Maybe forensic chemistry, we also test some narcotics or maybe blood alcohol. Those all kinds of tests also possible. In field of environmental, normally we are testing like agricultural chemistry, maybe some pesticides analysis, herbal analysis like herbicides or maybe phenols. In the clinical mechanics, normally medicines we are using for the checking of different extractions or different metabolisms of the APIs, normally estrogen and other analysis as well. So these are different scope of the HPLC. So what is all about? So obviously this slide is not for you because you know very well how to load the stationary phase, how to load the sample. Only depend on the RF value that get fractionated, and each and every fraction is now getting separated in the different component depend on your RF value and depend on your time. So this concept is used in the HPLC itself. So this is all about the HPLC, the reservoir bottles, the detectors and everything. So those kind of analysis can be possible with the help of this technique. So how it works basically. So normally the solvent reservoir or the mobile phase is kept over here and it goes towards the pump. So I'm just explaining the flow diagram of HPLC, how the flow diagram help to analyze the sample. So the mobile phase reservoir, we are keeping the mobile phase in the reservoir. The mobile phase come in the pump by acting as a pump, as either it's a binary, quaternary. That's all depend on the we'll let you know what is the different types of pump as well. So after pumping, it goes towards the column, and before the mobile phase goes towards the column, there is one injector is there. So we called as a sampler or at a sampler. So we need to inject that sample in the mobile phase, which mobile phase is passing towards the HPLC column. And after passing from the column, it goes towards the detector, and it just having some signal that gives the output of the computer data detector. And obviously the remaining solvent will goes to the waste. So how it works basically. So example, it's a simple flow diagram showing the two different solvents reservoirs having the two different mobile phase, and pump is here. And after pumping it gets mixed. So say example, it's an injector, and one column is placed over here. So, and it finally goes to detector. So what happened over here? Normally the mobile phases are two phases. First one is a stationary and second one is a mobile phase. So stationary phase is your column like C18 or C8 or different silica columns are there. Mobile phase normally carrying the sample from the injector towards the column, and after that towards the detector. So say example, if I am injecting the column from the injector, what happens? This is the chromatogram which showing the intensity on the y-axis and x-axis showing the time. So after injecting the sample, this is the first trial. So sample is coming from injector towards the column. And if you see on the top of the right hand side, there is one line started to building it. So this is your total dead volume. Dead volume is starting from injector to the column inlet, and as the sample passes towards the column, it as the sample passing towards the column, after getting the sample on the you may say column, which is a C18 or C8, it is an adsorption technique. Everybody knows that it's adsorption technique. So depend on the RF value and depend on the adsorption activity, that band gets separated. Okay, and as that bands get separated, almost there are 4 to 5, almost 1 2 3 4 5 6 bands are there. That means 6 different components are there in a given mixture. So depend on the polarity and depend on the mobile phase, whatever we are going to use, those peak are getting separated. And now after separation, the first peak, which is either polar or non-polar, depend on the mobile phase, it goes toward the detector. And if you see, this is called as the retention time. So first retention time, it's showing the peak at the first interval. So this is the first peak, now second, now third component, fourth, fifth, like that. So it just giving a tentative diagram about the HPLC. Now coming to the main topic, some important terms which are used in the chromatography is a chromatogram. Obviously everybody knows whatever there in the previous slide, which shows the intensity versus the time, is a chromatogram. Okay, and the mobile phase. The mobile phase means the liquid that moves the solute through the column in case of HPLC. So stationary phase, either the C8, C18, or maybe different kinds of columns which are normally used to separate the different components. Then yeah, the peak means it's a visual presentation basically in the chromatogram. Depend on the response, which is detector response. It gives the electric signal, and that we corresponds to the peak. Now retention time, the time actually taken after the injection up to the peak coming up to the detector. So whole time we call it as retention time, as it retain on a column. So how much time that component is retained on a column, we define as a retention time, and its whole depend on its polarity. Okay, so we'll come to the how it's going to play, how polarity will define what is actually, how to select the mobile phase, how to select the column. We in coming slide you will definitely let me know. So there are two purposes. First one is qualitative and second is quantification. So if you're talking about the qualitative, so qualitative means only identifying of a component. That's it from the mixture. How many components are there in the mixture? I'm just identifying it. That is called as qualitative analysis. But if you want to prove actual percentage of a component with respect to standard, so determine the amount or proportion of a component we called as quantification. So for the quantification, basically we are using these standards for as a reference, or for quantification we are using a specific quantity of a sample before loading on a column. So these are basic separations. Normally we are looking for the retention time, or selectivity is also important, efficiency, peak shape, resolution. Those are the basic phenomena which need to improve during the method development part. So this is the basic of separation technique. Now, if you see, it's a chromatogram. So as a time taken after injection up to the column, it's a dead volume. So after that, it's showing some notch over here, either from methanol or ACN. So this is the time we called as TR. It's a retention time. This is the area of that peak, and this is the height, basically we called as H, when it's coming to the in the power of signal. So what are the different separation mechanisms normally we are preferring? So components, so this is the column. When component, a mixture of component are passing through that column, normally depend on the polarity that component elute first or elute later. So for example, this is the red and blue. So if I'm talking about the reverse chromatography, reverse type chromatography, so obviously in this scenario there are two types of mechanism, basically reverse and normal. So if it is a reverse, that means polar peak elute first and then non-polar peak elutes. So non-polar component retain on more time on a bed of the Silla moiety. So how it's actually happen? So normally, if you just take an intersect of the column and having the stationary phase, so there is a strong interaction between the stationary phase and the non-polar moiety. Normally, due to the difference of interactions, the polarity differs and the molecule moves in a different rate. Therefore, the separation happens. So that's why if it has a weak interaction, obviously it moves faster as compared to the non-polar moiety. And this is the basic technique or separation technique you may call it, or actually happen inside the column. It's wholly depend on adsorptivity, not absorptivity. So in separation mode, there are two different, or not two actually, there are different modes, but normally today as a time concern, I'm not going in detail about the all modes, but we are focusing a normal phase, reverse phase, ion chromatography, obviously some size exclusion as well, and affinity. So there are different separation modes. So what are those different separation modes? What is normal chromatography is all about? So example, stationary phase is polar in property and mobile phase is non-polar in property. Okay, so this combination we called as normal phase chromatography. So stationary phase is polar. That indicates that I'm using a polar column, and non-polar mobile phase like heptane, hexane, or other mobile phases to separate the components. Non-polar, or maybe the normal phase chromatography we prefer to use for the separation of isomers, diastereomers, not enantiomer. Actually, enantiomer can be separated with the help of the reverse phase as well, because I have a lot of data which proves that enantiomers are separated on the reverse phase. So this is all about the separation of the chiral molecules. Specifically for the chiral molecules, we prefer to use the normal phase chromatography. Yeah, this is obviously no one want to identify it. So the stationary phase are polar. That indicates the Silla moiety is there on the stationary phase. Just imagine this is a column only. So on column, we are putting different types of Silla. There are n number of Silla moiety, depend on the which type of columns you are using. I'm just taking only one Silla moiety. So this is a simple silica gel column. If you are going to add any component on a Silla, the phase changes. Now we call that cyano column. Cyano means what? We are just attaching a modifier to the silica gel, and we are attaching some cyano moiety over here. Then amino columns, like we are attaching some amino over here. Some diol columns are also there. So depend on your applications, if you want to analyze some sugar moiety, so amino columns are more preferable. If you use cyano molecules or maybe normal C18 molecules, so general mid-polar to polar moiety, so those contain cyano or silyl silica gel is also useful. If you are going to analyze some protein molecules, so at that time normally we are using the diol columns. So what are the different phases of normal phase chromatography? Normally, as I mentioned already, mobile phase are non-polar in moiety. So like hexane, benzene, MDC, chloroform. Okay, so carbon tetra chloride or THF. These are some non-polar mobile phase which we prefer to use while we are going to analyze on the normal phase chromatography. Now, coming to the reverse phase, yeah, but in the field of pharma, if you ask me that what are the which mobile phase we prefer a lot. So normally reverse phase chromatography in the pharma sector or any other sector, normally we prefer to have at least 90% or to 95% reverse phase chromatography, hardly 5 to 10% normal chromatography has been used. So just focus what is a reverse phase chromatography. In reverse phase chromatography, the stationary phase is non-polar in property and mobile phase is highly polar. Okay, so this combination we preferred as reverse phase, exactly opposite to the normal. Now, these are the separate or different columns depend on the moiety which is attached to the silyl group. We define as the ODS, octyl, butyl columns, or maybe TMS. Tetramethylselenium column was also there in the beginning of when HPLC started. The first column was TMS, then the cyanophenyl C4, C8, and now it's a C18 column is there. So what are different types and why we are prefer to use these different types of columns? So we'll see in couple of slides, in coming slides. But yeah, the mobile phase which we prefer to use is water or any kind of buffer with some concentration, and obviously some organic solvent to pass the solute or to pass the mixture of a component towards a detector. So organic solvents are either methanol, acetonitrile, THF in some constant. It's not a pure THF as such. We are using THF in hardly 5%, 10%. And then buffer like phosphate buffer, acetate buffer, some amino buffer depend on the pK value of a compound. So there is a huge role while we are selecting the buffers in method development. Okay, so the ratio of aqueous and organic solvent is much more important while we want to separate those analysis, those component on the using the chromatography. So what is the interaction basically happens inside the column? So I'm just focusing on the column at first, and after column we are going to focus on the actually impact of a different components on HPLC and how it works. So what is interaction? Normally, if it is a silica column, silica gel, and if it is a silyl moiety, if we attach a C18 carbon chain over here, it's become a C18 column. So it's a support particle, and this is a non-polar bonded molecule. That means it's a less polar. A is less polar, and B is more polar. So B, as it indicates, is a less polar, or you may say more polar, not less polar. So it has a very less interaction with silyl moiety. And as the A is a non-polar, it get absorbed on the silyl moiety and C18 column, C18 structure. So more hydrophobic analyte, that means less polar, get attracted and spend more time. Okay. On the other hand, the polar moiety spend less time on the silica gel. So what happens in that scenario? For example, if you check the hydrophobicity, there are two different components. I'm taking example of carbon chain, and second is one of the aromatic component. And there are different components in the other samples as well. For example, it's acid or maybe ammonia or maybe some of the hydroxyl component. So in first scenario, it's a hydrophobicity is stronger, right? And second one, hydrophobicity is weaker. That means if hydrophobicity is stronger, that indicates it's a highly non-polar compared to this peak. So acid, amine, or alcohol, those are the highly polar component as compared with benzene or any carbon chain aliphatic components. So now you will get the fair idea if it is a silyl moiety and if I am attaching the ODS, that is C18 column, octadecylsilane, by the way, ODS means octadecylsilane, C18. So if I have two different components, one is a phenol and second one, phenol is there, but apart from that, there are two different moieties. Like, if you are attaching CS3 and there is a strong interaction between CS3 and C18 as compared to OH. So it shows a weaker interaction, and that's why it elute first as compared to second component. I'm talking about a reverse phase chromatography. So polar component elute first and then non-polar elute, depend on the retention time. That means how much time it's going to retain on that particular column. So what are the different factors affecting to the separation? So the first factor is the solvent, mobile phase. So I'm just taking an example of one standard like methyl ethyl propyl or maybe butyl paraben and a mixture of those parabens, and see the first picture of water and methanol 80-20 ratio. I'm just focusing on isotopic mode. So water is 20% and methanol is 80%. And the last one, water is 40% and methanol is 60%. As I increase the concentration of water or vice versa, as I decrease the concentration of methanol, I'm getting the better picture. By the way, I'm not changing anything, not the conditions, not the column, only I'm changing the solvent polarity. Okay, if I increase the solvent polarity, I'm getting the better separation. So this is the first factor. Second factor, effect of the stationary phase. So in case of C4, C8 and C18, there are three different mobile phases or three different columns are there in the market. So as carbon chain is more, you will get more area for the compound to have some adsorption or to have some strong bond to correlate with as compared with C8 and C4. So C4, obviously it's a weak because I have very less space or less area for interaction. C8 is also a very less area for interaction. But yeah, each and every column chemistry has its own applications. It's not like that all column chemistry is happen only with ODS. It's not like that. Some of the sample which are highly polar are also we are using C8 column for the separation. Okay, so I just covered the impact of the mobile phase. Now impact of the stationary phase. For example, I'm keeping the analytical conditions as the same, water and methanol in the proportion of 70:30. Each and every parameter is same using the benzoate now. These are the methyl ethyl propyl or butyl benzoate. Initial column chemistry, TMS, tetramethylsilane. I'm injecting the same sample, using the same analytical condition on different column. I'm getting somewhat better resolution as compared to TMS, but as compared to TMS, I'm getting better resolution on ODS column like C18 column. So this is the impact of how much area we are going to provide for the particular sample to stay on the column front. Now, this slide is most important slide of this presentation. We call it as a polarity of a common organic solvent and functional. Whenever you are going to develop any kind of method, just follow this slide. For example, aliphatic hydrocarbons are highly nonpolar. Each and everybody knows that. And if you see the carboxylic acid, as it contains acid or it's a proton lover and proton donor. So this is the highly polar as compared to aliphatic hydrocarbon. In between, olefins, aromatic hydrocarbons, halides, sulfides, everything is coming in between. So when in terms of organic solvents, if you are comparing in terms of organic solvents, hexane is highly nonpolar as compared to water, right? So this slide will helpful for you to just start with the mobile phase, combination of mobile phase, and which is the functional group of my component. So this will help you to start your HPLC method development. Either I'm using water, either I am using acetonitrile, methanol, or need to use some non-polar component as well, some kind of hexane. So for the choice of MS, as I covered, there are different types. I'm not focusing on ion pairing right now because it is itself a huge topic. So okay, so in the mode itself, it's a normal phase, reverse phase, ion pair, and SFC. So or maybe size exclusion, not SFC. I'm talking about size exclusion chromatography. So normal phase, the solvent type is organic mixture, either isomer. I already explained you that isomers are normally some kind of molecules which are not possible or which are not soluble on the water or organic molecules. Those kind of analysis we can use a normal phase as your LC mode. If the molecule is neutral, non-ionized components and easily dissolved in water or organic mixture, it's easy to use reverse phase chromatography in that scenario. In the reverse phase as well, there are two types of chromatography. It's a highly polar moiety and somewhat, towards the non-polar moiety. So if I'm using a polar moiety or want to separate a polar moiety, but it's not possible because of the pK value of a compound. So today, obviously, I'm not going to ask any question on pK value. I'm not going to tell anything because it's pK value started from standard 12 itself, 11th. So as everybody aware is that pK value plays a huge role of selection of the pH of a mobile phase. So if the pK value we need to change, we need to introduce some ion pairing reagents in between so as to get proper ionized and get a proper adsorption on a column itself. So after formation of some complex, we call it as ion pairing reagent complex. And ion exchange chromatography is also there, either cation or anion exchange chromatography. It's already depend on inorganic ions or maybe some organic acids. And size exclusion chromatography, we are preferred in case of polymers like peptides or protein molecules, which are having a, you may say, gel permission chromatography, GPC. So those molecules, if having the molecular weight more than 20 kDa or maybe 40 kDa, in that scenario, we need to use the gel permission chromatography to identify what is the range of that particular proteins. I'm talking about a molecular weight of a protein. So in a broad perspective, if you see the scope of the HPLC, as I explained, ion chromatography, ion exchange, gel filtration, or GPC, or maybe the normal phase, reverse phase touching all orifice of all the component. So normally 80 to 90% of a chromatography is dominated by the reverse phase. Okay, so that's why I'm just focusing on reverse phase chromatography today, because each and every chromatography has its own challenges and own advantages. So what happens, we will see the instrumentation part, how it, instrument act as a HPLC. So there are two types of the configuration. The first one is isocratic and second one is gradient. But in the gradient itself, there are two different components, either low pressure or a high pressure. So what is low pressure and what is high pressure and what is isocratic? So isocratic, that means it's a premix mobile phase. I'm just passing through the column throughout the analysis. And gradient means I'm applying different concentration of either solvent or maybe say your organic phase with a stipulated time by giving some gradient program in the instrument. So in isocratic, in a simple way, if you say it's a mobile phase, simple system with one pump and one solvent reservoir, that's it. If you want to use a more solvent, we need to pre-mix it, either buffer or organic solvent like acetonitrile or methanol. We need to pre-mix it, and after that we just passing that pre-mix sample and passing through the column, and we will get the data. What is a low pressure gradient? So low pressure gradient means there are four reservoirs, four different mobile phase, and in the four different mobile phase, we are putting some solvents. One is either your mobile phase, either in the basic or acidic condition, then acetonitrile, methanol, water, whatever you want to put, depend on your mobile phase development, method development. and low pressure gradient wall is there. So in the low pressure, the most important point is that mixing done before pumping. So in the low pressure, there is only one pump is there. Mixing of the mobile phase happen before the pumping. The mixing of a mobile phase, we are normally putting the gradient program in the software. So according to the software program, the mixing is happening over here in the low pressure gradient wall, and one line degasser is necessary because after mixing, normally what happens, there is a formation of some exothermic reactions over here with the help of water, acetonitrile. Obviously, there is some formation of exothermic reaction. That's why there is a formation of air bubble during after the mixing and passing towards the pump. So we are putting one degasser which helps to reduce these kind of fluctuations. So degasser is compulsory. Now, as I explained in the reverse low pressure, mixing happen before pump, but in the high pressure, mixing happen after pumping. So there are three different pumps and three different reservoir. The pumping done for each and every reservoir, it's a different pump. After pumping, it goes to the mixer. In that scenario, online degassing it may not be such a critical because after pumping, if it goes to the whatever the evaporation or whatever the exothermic reaction will take place, the time is more. It's not takes place over here, it's takes place over here, but obviously air bubbles and everything is gone in the pump, so it will not play any huge role in the high pressure gradient, but it has a excellent gradient accuracy. 2 to 3 pumps are required and mixing done after the pumping. So these are the two major difference between high pressure and low pressure. Coming to the detector part, if I focus on detectors of the HPLC, normally we have the different detectors like UV visible, obviously PD detector is also there. So I will cover each and every detector: RF, RID, ELSD, MS. So these are different types of detectors we are prefer to use when we are clubbing this HPLC as such. That whole depend on your applications, which types of applications you are playing with on the HPLC. So obviously, once again, the basics of whole HPLC, Lambert-Spear's law, right? So in the Lambert-Spear's law, everybody knows that whatever the absorption which is obtained on the chromatogram, it's directly proportional to the molar absorptivity and the concentration of analyte. If I'm keeping the path length of the flow cell as the same, okay, so your concentration, in simple language, if you have the lower concentration, you will get the lower absorptivity. If you have the higher concentration, you will get the higher absorbance. Okay, that is the Lambert-Spear's law. And on the principle of this Lambert-Spear's law, UV visible will act as a detector. Now, what are the advantages and what are the disadvantages of the detector? You are going to say disadvantage? Yes, it's having only one disadvantage, but obviously it is more sensitive as compared to any other detector in the category of, if you compare with PDA and UV, obviously UV is more sensitive. Then relative robustness of the temperature and flow rate change as well. It's a robust if you're changing the flow rate or if you change the temperature, it will not having any strong impact as such on the detection level. And obviously UV is a gradient compatible. So some of the detectors in the HPLC are not gradient compatible, but this detector UV is a gradient compatible. The only disadvantage of this UV is, only compounds with UV and visible absorption can be detected. Okay, so if the compound is UV active, then and then only this detector can be useful. The additional feature is that with respect to the wavelength, you may have the two different wavelength modes. Example, if I have the wavelength showing 210 and 260, so both wavelength mode can be possible. Wavelength time program also important, and wavelength scan mode can be possible with the help of these detectors. So these are some advantage of UV. If I'm comparing with PD detector, so what happened in the PD detector? This is the lamp. Now the range of the PDA is starting from 190 to 800 nanometer. So if the compound is unknown, as in the previous session, sir also told that if the compound is unknown, PDA will help us to move ahead with that unknown molecule as well, and it will give you the exact lambda max of a compound if you don't know the lambda max as well. So that is the advantage of PDA. So it passes. This is the sample cell. Normally, sample is passing through the sample cell. We call it as a flow cell, and we are keeping flow cell near to the D2 lamp. And after, as rays passing from the D2 or W2 lamp. So the first one is a D2. So D2 work as in the range of 190 to 400, and W work as a range from 400 or maybe 180 to 800 like that. Sorry, 380 to 800 W2. Now it passing on a grating and showing a different elemental photodiode array with almost 1024 elements are there. Each element detects one absorbance at wavelength, at one wavelength. So it will give you the more accuracy as compared to any other detector presence on the in and with regards of the wavelength. So the advantage of this detector, say example, I'm just putting a 3D dimension, just focus on this red chromatogram. So red one is a chromatogram. You're just focusing on the peak number 1, 2, and 3. So this is your chromatogram. And if I'm putting the absorption over here and the wavelength over here, so time, absorption on the left hand side, and wavelength putting over here. So the two spectrum of two different component. So it's showing some wavelength as peak comes, it's showing some wavelength as such. So these are the two different wavelength with respect to two different peak. So the PDA help us to provide the more accurate data. In this slide, you may get more clarity about the what I want to say. Say example, this is the time starting from 0 minute to maybe 4 minute. Okay, and these are some wavelengths starting from 200 or 190 to maybe 300. So as I inject the sample on the column, either before 1 minute or 2 minutes, there is one single peak showing the wavelength of 200. After that, if I'm getting a second peak at the maybe 2.3 or 2.4 minute, I'm getting two different wavelengths, one at 200, second at maybe 260. Third also, I'm getting the two different wavelengths for this third peak. So depend on the lambda max of your compound, either it's a pure or non-pure or having some mixture of a compound in that peak. This PDA will help you to resolve those impurities. So we act as we called as a peak purity of a peak purity concept. I will elaborate that concept in next slide. But how what are different applications of PDA to identify? For example, these are some acids like azelaic acid or maybe benzoic and nitrobenzoic acid. So normally, if I use the concentration of ACN and water as 70 to 30%, so 30% ACN and 70% water. And if I want to slow down, if I want to resolve these molecules, three different components very well. So what I did, I just decrease the concentration of organic and increase the concentration of aqueous, right? So starting from 30% to 15%, I reduce the concentration of organic phase starting from 30 to 15. Now I'm expecting the peak of first peak should be alizic acid, second one is benzoic acid, and third one is nitrobenzoic acid as compared to this. But if you see the PDA spectra, or if you see the PDA application or the spectra of a component, so this is 70-30 and this is 15-85 ratio. First one is azelaic acid, second one is a benzoic, third one is a nitrobenzoic. But in a second scenario, the data or the peak completely flip from the peak first to second, and this actually come first from benzoic, second to first. How we are able to identify? With the help of the spectral analysis using the PDA, because each and every component has its own spectra in the, you may call it as a fingerprint region. So it's better word to have it. So each and every component has its own spectra corresponds to the lambda max of the compound, and that's very easy to identify. The peak number 1 corresponds to the benzoic acid and peak number 2 corresponds to the azelaic acid if I use this type of gradient. So one more advantage is about the peak purity checking. So example, salicylic acid, maybe I'm using the acetyl salicylic acid and this is the standard compound. And if I injected the sample and just make the overlay of the spectral component, so standard is this one, but sample showing some of the unknown peak over here in the spectra. That indicates that main peak contains some impurity in it. So to check the peak purity, PDA is also useful. If the peak is present in the tailing or maybe fronting, or it is getting completely merged with the peak, the PDA help us to get it identify. And if I get identification that the main API contains some of the impurity, I also need to develop some method which help me to resolve these kinds of impurities. So what are the advantage of PDA? So PDA normally used for analysis of simultaneous of many wavelength, as many as possible, starting from 190 to 800. UV visible spectra are useful for the compound identification, checking peak purity, as well as the optimized absorbance. Okay, the last is relative rubberness of the temperature with respect to the flow, and obviously it's compatible with gradient illusion. And one more feature normally we prefer to have is that we may use to store the library of a spectrum. That means I used, for example, acetophenol, or I'm going to use the metformin or ticlopidine, or maybe ciprofloxacin as a component, and I just want to store that spectrum as a library. When I compare those components with the different unknown components, I need to check or overlay the library and just checking the ticlopidine is present or not, or metformin is there or not, or maybe ibuprofen is there or not. So this spectrum will help me to have the identification test as well when you are using PDA. Now, coming to the ion pairing chromatography. So what actually ion pairing chromatography is doing? So obviously I'm going a little fast, but I need to cover whole topics. So yeah, in the reverse phase chromatography, normally if we inject some sample and if it is not stored on a silyle moiety, it elutes faster. So it's highly polar in nature. Okay, having very weak interaction with C18 column. So for that purpose, we are going to couple those sample with some of the reagents which actually shows the ion pairing on it. So what are those reagents? Those anion compounds are maybe TBA, normally we call as tetra butyl ammonium hydroxide. Then maybe some of the salt we called as sulfonic acids, sodium salt. So either it's a butanic or maybe pentanic or hexane. They depend on the carbon chain. We are having the cation exchange ion pairing reagents. So normally we are using heptane or hexane sulfonic acid, sodium salt, to have to increase the polarity of a component. How those are going to play a role while developing a method development or any kind of development? So I'm taking example of the two moieties like benzoic acid or toluic acid. So if I'm using column of C18 and the mobile phase is obviously 50-50 water and methanol, and tetrabutyl ammonium hydroxide has been used. So in that ion pairing reagent, nicotinic acid or maybe biotin or maybe folic acid, caffeine, thiamine, those different components are there, but I need to separate each and every component. For that purpose, I'm using sodium octane sulfonate as an ion pairing reagent. So as to that will help me to resolve each and every component in a slow manner. But it also having some advantages, also having some disadvantages. So yeah, this is all about the ion pairing chromatography is all about. Just define the pK value of 4.5, and if I am using the pK of 6. So what is the impact of ion pairing reagent on a solvent? So normally, hexane sulfonate acids, example, I am using hexane sulfonic acid and pentane sulfonic acid. So its whole depend on which type of ion pair chromatogram or ion pairing agent you are going to use with the help of mobile phase. But What are the disadvantages basically? Advantage is, yes, we have the ion pairing selection. We are playing with pH. We are having the selectivity for the concentration. Selectivity can be improved, definitely improved, but same time, disadvantage is that equilibration time is much more as compared to the reverse phase chromatography. Dedicated column must, normally we are recommend to use the dedicated column for ion chromatography. This column cannot be used for the other analysis or other molecular analysis as it contains some ion pairing reagent. We need to wash the column thoroughly before analyzing other analysis of same ion pairing reagent as well. So there are different column chemistry need to participate with, play with the ion pairing. So this column chemistry or ion pairing chromatography is not as much familiar or not as much popular as compared with other chromatography section. So obviously we require some washing reagents as well. So modifier, if you use some modifier or TBA, then we require to wash the column with the help of mobile phase or with the help of different washing solvents, or maybe phosphoric acid including 100 millimolar sodium perchlorate and methanol 50-50. There are two types of ion chromatography basically, anion and cation exchange. So obviously the name indicates if it is an anion moiety sample having carrying the negative charge, we need to create some anion exchanger over here, and that's we called as anion exchange chromatography. Okay, and a reverse about the cation exchange. With silica or the resin, we need to form a cation exchanger over here, and that forms with the help of ion exchange with whatever we are using, like sulfone, hexane, or maybe ammonium hectane column chemistry. So ion exchange can be used typically with some of the organic cations or inorganic anions, or maybe organic acids. Peptide and protein molecules in biological field, normally we used. Now reverse phase chromatography or amino acid analysis, also we are using reverse phase chromatography with some modifiers. So those applications are also with me. So coming to the other detector, just covered the PDA and UV. Apart from that, there are other detectors. If the compound is not UV active, or you may say it does not contain any chromophore in it. So in that scenario, we need to use other detector apart from the UV and PDA. So there are two detectors specifically. One is RI detector, which has refractive index, and second one is ELS detector. So it's called, the name itself indicates it's evaporating light scattering detector. So RI means, but the disadvantage of RI is I'm not able to use the gradient mode on RI, and it's not compatible with gradient. But on ELSD, it's gradient mode compatible. What is the function in the ELSD itself? When the component coming from the column of HPLC, nitrogen has been used as a nebulizer gas in this entering tube. After that, there is a drifting tube present over here. It drift, and after that it goes towards the detection where the scattering is happening. So over here, light source has been there, and with the help of light source, I'm just applying the light source, and depend on the targeted molecule, the light gets scattered, and that scattered angle has been measured in the ELSD. So that's what it's called as light evaporating light scattering detector. What happens actually inside the ELSD? So whenever any kind of mobile phase is passing from the after the column inside the detector, over here some of the nitrogen gas has been provided so as to get the nebulization to just form this formation of spray, nothing else. So after formation of spray, some of the higher molecule or maybe the droplet getting drain out from here, but some of the small particle size drops are moving along with that nitrogen gas and moves towards the drifting coil. So in the drifting coil, we are applying a certain temperature where the surface tension of that particular droplet get increased, and at least it form the blast, and whatever the vapors are getting formed, all vapors get evaporated, and only some of the component of a molecule passing towards the detector, and we applied a light source. That light source passing through the beam and just passes through that particle, the light get diffracted. Okay, so basically that's why it gets scattered, and that scattered angle counted on the this counter, and we get the electric peak over there. So the first step is nebulization. Normally nebulization happen with the help of mixture of the nitrogen gas, formation of the droplet. Second one is modification or the mobile phase evaporation. As I mentioned, mobile phase is there along with single each and every droplet. So mobile phase get evaporated. Dried sample particle remain inside, and then it goes to a detector. As light passes from this lamp, it actually pass on that drop or the photoiode sample particle and gets scattered, and we are just counting the scattered angle, and with the help of that we are just getting the peak of the component with the help of this mechanism. Now obviously LSD is a universal detector. It's highly sensitive as compared to RI, and it's a gradient compatible. That's why everybody using it if your compounds are non-UV active. Better selectivity, obviously it has a better selectivity, and no solvent from the peak. If you compare RI and LSD, these are the two chromatogram. I'm just putting, see the sensitivity of the peak. For just correction, I'm just making peak 1, 2, 3 with the same RT, but for just simple understanding, I'm just moved that chromatogram just near to the other one. So if the RT is same, normally the sensitivity, if you see with RI and LSD, it's a huge difference. Now the last detector is MS. So on this detector, actually, I'm not going to talk much today. I will cover this whole detector and whole mechanism applications on coming session, but I think it's on tomorrow. So I'm just focusing this mechanism as per as to our today's session is concerned. So what are the designing basically in MS? This is the atmospheric pressure and the high under. There are two vacuums. One is high pressure and one is low pressure vacuum inside it. So roughing pump is there, and apart from there are some turbo pumps are also there. So in roughing pump we created the vacuum up to e raised to 2 torr, and in turbo pump the vacuum is almost e raised to -7 torr. So it's almost a very high vacuum. So when molecule is passing from HPLC to the some probe of the MS, it doesn't have any content of, it doesn't in the having any atmospheric you may say vacuum inside it. So it's just atmosphere over here, and as mobile phase passes from the detector of the API probe, as it comes out, it also contains one gas which used for the nebulization of a component. And after nebulization, the gas normally we prefer to use is a nitrogen gas. That component get nebulized, and as they forms a droplet, those droplets are getting evaporated. We are applying temperature over here, and that moiety either in the form of positive or negative ion passes towards the detector and detects the mass of a particular component depend on the RT of that molecule. So you may either quantify or qualify it. So this is the phenomena. Normally there are two probe, ES and APCI. I will cover this whole mechanics tomorrow with ES and APCI. But this how it takes place when liquid sample coming from this tube, as it passes, apart from that we are applying some nebulization gas and also some voltage. So just applying gas, it forms some droplet, and evaporation of the solvent takes place as we are applying some temperature over here, and that temperature is almost more than 200 or maybe 150 to 200 degrees Celsius. Those all solvents get evaporated and only decrystallization get formation, and after that the ion passes inside the MS to identify the mass. And second case, we are applying APCI. Normally it's called as non-polar For the non-polar moiety, we are using APCI. For mid-polar and polar moiety, we are using ESI as a probe. Okay, so these are some applications depend on the your characterizations. Example, if you want to characterize like protein or some kind of natural products, or maybe some of the drugs like drug metabolism. If I want to analyze some drug metabolism of maybe metformin or maybe ticlopidine or maybe some of the kinds of what do you call it as maybe paracetamol. So we don't know anything. So there are different types of API, maybe fluticasone propionate as well. So we have lots of API where we just want to identify the metabolism of a drug, where we are using this kind of ESI probe. Maybe some peptide moiety analysis, protein moiety analysis. If you want to do the pesticide analysis or steroid analysis, we are using APCI, as pesticides are almost on a non-polar side, highly non-polar as compared to peptides or proteins. So depend on the polarity, those kinds of, the benefit of the LCMS. Normally there are two-dimensional chromatography. Basically, we are getting peak as well as the mass of a peak. If you see, there are three peaks. First one, if you say ABC, so actually on chromatogram, HPLC chromatogram, you are able to see three peaks, 1, 2, and 3. But in the middle peak, there are two different components having the two different development part when it comes into the ppb level or maybe ppm level when you want to quantify or qualify. And also, if you want to determine the molecular weight, this is one of the kind technique to have it. There are different ms like single quadruple, triple quadruple, TOF, those kind of techniques are there. Now, how to select the detector depend on the compound which you are using, because there are huge number of detector, but obviously the question come in the mind that which detector is more preferable, either UV or maybe LSD or maybe RI or maybe RF. What kind of detectors are those? And if the compound is fluorescent in nature or fused with a ring or maybe highly conjugated planar, then fluorescent detector is useful. If the compound is having chromophore, obviously, and if having such as aromatic ring or multiple alternative double bond, UV or PDA range is useful, detector is useful. But if I don't know the lambda mass of a compound, I will definitely go with PDA. Then for the CD charged compounds, if those are the inorganic ions or maybe organic ions, normally we use CAD detectors. Then the compounds which are oxidized, like quinone or maybe amines, we used ECD detectors. Though now this detector is not used as such longer. That's why I'm not explaining today. So RI and LSD. So RI normally we are using for some sugar moiety, saccharides, or maybe some of the analysis like polymers or different analysis we are using the RI detector if it is isotopic only. But if it is gradient, then definitely yes, we need to use the LSD as a detector apart from any other detector if the compound is UV inactive. Okay, so if all those detectors are there, then what about this comparison? How, on which basis I'm going to compare it? So I'm just making one slide where you will get the comparison of each and everything, like typical limits of detection. I'm not going in detail about the quantification limit. It's wholly depend on the absorption capacity, and molecule to molecule it may get vary, but obviously we are just taking with some standard. So with the help of that, PV, UV, and PDA up to 1 ppb in some extent, we also worked extensively. Yes, it's a gradient compatible fluorescence in 10 ppb as compared with RI, it's 100 ppb. So if you see the RI as well as in RID and RF, so RF is more sensitive as compared to RI, and obviously ELSD is more sensitive as compared to RI and equal to RF. And MS, you may go up to ppb or maybe ppt, depend on which kind of MS you are going to use. So yes, there are some red flag over here. RI and CCD are not gradient compatible. In this scenario, ACD, then conductivity and refractive index these T detectors, we need to use isochromatic mode only. So that is a limitation when you are going to develop any kind of method. So coming to the HPLC, how it looks and how it's look like. So there is one I series where this is all about the I series plus HPLC. Nowadays, HPLC has been modified like anything, because now AI and IoT concept has been came. So it's artificial, but we call it as analytical intelligence. And so analytical intelligence means it starts from auto startup to auto shutdown. Everything has been done with the help of HPLC. So obviously, it's you can hook up any detector with it. Advanced intelligence support to multiple detectors, either LSD, UV, PDA, whatever detector you want to hook. It's very easy to hook up. Then remotely access is possible now. Nowadays, it's as a human, error, or to avoid the contamination, we are preferring to use as a remote functionality. Eco-friendly, and eco-friendly means it's having saving the power almost 95%, as it has the function of auto startup to auto shutdown. Obviously, these are some detectors which are preferable to use, like fluorescence, RID, or maybe LSD, and smart flow is also there. So when any analysis come, normally we need to, what is the actual role or what is the flow diagram of the analysis? In simple language, if someone is coming in the morning in the lab, so you need to start the instrument, purge it, then we need to keep some time for column equilibration, stabilization, for the baseline. Then after that, we started the analysis. So almost 1.5 hour to 2 hour will be required for this purpose only. And after that, the analysis will start. And after sample completion, the analysis finished, and then cool down and pump off and shut down. So this is the normal flow. But what it contains basically, if we given some program, automatically it will start, or it will wake up in the morning at 6 o'clock, as you just having using the software, and it will wake up in the morning. It will do the startup. It will purge the system. It will have the all the equilibration of the column, stabilization of the baseline. And if you put the sample yesterday evening, then also sample will start in the morning. And after completion of whole analysis, it get shut in complete manner. It's not like only pump getting off or only lamp is getting off. Whole instrument has been shut down, and that's why we are saving the energy, and 95% of the energy. So these are some of the bullet points normally of the our Simatsu model. Now we call it as AI series plus. One more advantage is that of this particular model is it's coming with some functionality. Normally we called as the high throughput functionality. So what is high throughput? Now, if, for example, I want to prepare a solution of 10 ppm from 100 ppm, so obviously we need to prepare from outside. So human error also include, maybe some of the pipette issues are there, some of the other parameter is playing the role while dilution or spiking or co-injection. So all these functions are happening inside the auto sampler itself. It is a completely automatic. So it's we called as auto treatment functions. So this is also possible. And nowadays, everybody prefer like 21 CFR compliance software, if you ask, or 21 CFR in the concept of the pharma sector, if you ask. Those parameters are very crucial. Zero carryover, as much as 0.0025% carryover is there. Then high polar or low polar compounds on the same system you can use. So you may use the hexane, you may use the water, you may use the any other detector. Just need to while transferring from one phase to another, we need a thorough cleaning. That's it. So that is some of the cutting edge technology we have. And if we see about the future, so the future is some time, normally amino acid analysis we are going to prefer on the, now since last 3 to 4 years, amino acid analysis has been changed, or the direction like anything. So for amino acid, we need to do the pre-column derivatization or the post-column derivatization for identification. As all amino acids are normally non-UV active, we need to identify after derivatization on a fluorescence detector. So everything is happen on the needle itself. If you see, the vial position as well as the volume capacity I have mentioned over here. So by getting the air gap, I'm just mixing the MPa, OPA sample and the different reagent for mixing purpose, and each and everything get mixed, pre-mixed on a vial and get you may say pre-column derivatization. Derivatization happened in one of the vial, and that sample has been taken for the injection. So it avoid the all human errors. So almost 3 to 4 hours for the baseline check, stabilization, mobile phase, sample treatment, all can be avoided with the help of these automatic sample treatment functions. And this is the future of a lab. You may say there are no HPLC as such, and this is a practical view of one of the pharma sector. So there is no PC, it's a complete PC free lab. So this is all about the HPLC. And yeah, so if you if we want to talk about the different applications, I just want to share some applications as well. Just a moment, I will stop the screen and share the other screen for the different applications. Or before that, I think it's better to have some interaction with you. So what you understood, or do you have anything in your mind, or do you have any questions regarding the basics of the HPLC or any detector? Let me know. So that will be easy for me to move ahead. Then I will cover some applications. I will just slide some applications, 3 to 4 slides.
Sorry, sir.
Hello.
Hello.
Yes, I'm Professor Naule from Government College of Pharmacy, Ratnagiri. Just I want
Okay.
to ask one question is that whenever there is an MS detector for the HPLC, at that time, how they manage the sampling system? Because there are lot many coming from the column, and whole thing cannot go into the MS. Because
Right. Great.
as far as the working pattern of MS is concerned, that we can push all the things in UV detector, but not in MS.
Yeah,
Because
absolutely
working of MS
right.
is a little bit slower. So how that sample continuous flowing of it is monitored?
Yeah, absolutely right. And it's the right question basically. Whenever we are talking about the MS, it's on a very high sensitivity detector in any other detector. And whatever the flow rate we are applying to the HPLC, it is not directly going to pass on MS. We are using splitter in between. So it is exactly half of your actual flow. The maximum limit of the flow rate is 0.5 ml per minute for MS. And after that, even if we applied 0.5%, 0.5 ml in the MS, so before that there is one ion chamber. So what happens in ionization chamber, we applied certain temperature to ionize as well as to vaporize the mobile phase, whatever is coming from HPLC. So after vaporization of a mobile phase, we are pushing that ions into the MS. So indirectly, any kind of solvent are not allowed to enter in the MS detector. Okay, so we need to optimize the MS temperature as well as different kinds of voltages before that molecule is passing towards the MS. So in brief, I will cover how it works in actually in tomorrow's session. So it's basically on the MS. So I will definitely, that will help you a lot.
Yes, sir. Just one more, just out of curiosity, from your answer only, I got that question. That if that solvent content dissolve matters, like we are adding the buffers and all these things, so are they going directly into the MS then?
No, no buffer or no any other solvent will go inside the MS. Before MS, there is one probe we called as ion source or ion guide. Or whenever any mobile phase is coming to that probe, okay, we called as ion source. So ion source is in the atmospheric pressure. Just a moment, I will show you once again that slide, that will be better. So whenever we are talking about the ion source, normally what happens, there are two different pressure. One is atmospheric pressure and other one is high under vacuum pressure. So this is the vacuum pressure, right? So MS starts from here onwards. From here onwards, MS starts actually. If it is a single chord or triple chord, that's another part, but MS starts from here. whatever the ionization or whatever the evaporation happens, it happens in atmospheric pressure. And we are taking care of the solvent. For example, if I'm using a water or some compatible solvent to MS, as you mentioned, there are different buffers. So normally there are do's and don'ts in MS. So certain buffers are not allowed to use in MS for analyzing, because those are actually hamper the analysis and decrease the concentration of the my actual API. So like formic acid or maybe some of the acetic acid, that those of the acids has been used like in the percentage of 0.1, 0.2. So though all acids are getting evaporated over here, all solvents are getting evaporated over here, and only analyte containing the charged ion allowed to pass in atmospheric pressure. I think, hope you are clear, sir.
Yes, definitely. Thank you very much.
Yeah.
Thank you. Thanks a lot.
So any other questions do we? Yes, sir.
Anyone have the questions? Participants, please. Okay, if anyone don't have the questions, will we go for the vote of thanks? So I think no need to propose a vote of thanks, because Kokate sir is with us tomorrow with new topic. As a formality of today's session to complete, I propose the vote of thanks on the behalf of Government College of Pharmacy, Karad. I express my sincere thanks to the Kokate sir for enlightening to our participants on the basics of chromatography and different detectors, how to use and what is the principle in the various detectors. Sir has explained very clearly. So with this, I once again express my sincere thanks for giving your valuable time with us. So now I request to Sarade sir to give a certificate of appreciation in electronic form.
Sir, please accept the certificate in the virtual form. We'll send you the
Thank
mail.
you. Thank you very much.
Yes, thank you very much. Thank you very much, Rahul sir.
Thank you, sir.
Thank you, sir. Thank you, Rahul sir.
Yes, sorry, sir.
Yes, sir. Now over to the Osmani sir.
Good afternoon, all. Morning session is over. So we will come again at 2:30 o'clock.
Participants are requested to join by 2:25 at least. Yes, thank you very much.
There will be two interesting sessions of Mr. R. Girijan and Sri
Venkatesh. Okay, thank you very much. Okay, see you after lunch.